Welcome to the first grand rounds in a new decade. Thank you very much for being here. It's a pleasure to introduce Saar I Gill. Saar received his MD and PhD in Australia, as you'll be aware in a moment. He's Australian, and he was telling me last night that one of the advantages of having a combined degree is that some of the work that he's done, he's actually done in the lab. He's made constructs, he's put them into cells, and then he's put them into people. So it's the entire soup to nuts program, which I think is fantastic. So as I mentioned, Saar got his MD and PhD degrees in Australia and Melbourne, then did a residency in internal medicine before coming to the States, where he joined Rob Nagrin's group at Stanford working on NK cell immunotherapy and GVHD. He then went to UPenn to do a fellowship in BMT in 2011, and then joined the faculty there in 2015. Saar is an interesting fellow. He has over a dozen patents. He's published in a number of high-impact journals, such as Cell and Blood. He's got a recent paper in Cancer Discovery, Journal of Clinical Oncology, and he's also started 2 companies. So he's an energetic fellow, a rising star in the field of cellular immunotherapy, and we're really happy to have him to present our inaugural grand rounds for 2020. Thank you.
Thanks for the kind introduction, Chris. So CAR T cells 2020, I initially was planning to make this rather a high-level overview, and I will spend the first half of the talk as a high-level overview of what I see as the exciting as well as evolution of CAR T cells, as well as their challenges. And the second half of the talk I'll spend sharing with you a paper that was just accepted last week to Cancer Discovery. So the educational aims really, as I mentioned, are to describe the clinical outcomes of CAR T cells in hematologic malignancies, where really that's where the majority of the effort has been to date, the successes, I should say. And very briefly in one slide, unfortunately, to describe the clinical outcomes in solid tumors, and that is probably a topic for another day. But something that we really need to work very hard on. And finally, to describe a novel mechanism of CAR T cell failure, which I think is really, well, I think it's interesting, but look forward to seeing what you do about it. Here's a very high-level overview of the complete response rates of hematologic malignancies to CAR T cells, and I would point out that generally, as one would expect from early-phase clinical trials, these are patients with very, very, very advanced diseases. And so across the board, you can see in ALL, a complete response rates of around 80 to 90%, and the vast majority of those are minimal residual disease negative, or measurable residual disease negative, so very deep responses. What I like about the field of CAR T cells, as a slight aside, is that the results are reproducible, the clinical results are reproducible across different centers, and so to me that means they're real, not just a flash in the pan. Non-Hodgkin lymphoma across the board, and this is for City19 targeting with CAR T cells, we see lower response rates of 40 to 50%. Let's say 40% I think is where we're really coalescing around. And in CLL, which is some of my clinical trials have been in CLL, we see somewhat lower complete response rates. And myeloma, I think the percentages there are maybe a little bit overestimated, because here we're talking about what the myeloma docs call VGPR plus CR, so very good partial response plus complete response. It's not the same thing in my mind. We need to aim for very, very deep responses, and that's not what those numbers are. So, but a really critical question, beyond the initial enthusiasm of the first few years where we saw these responses, and the initial enthusiasm that as a treating clinician I have for a patient who achieves a complete response who previously had refractory disease, are what is the durability of this response? And I think what has been quite remarkable, here are 7 different papers from different groups, including our group, Children's Hospital, so Penn. This is a multi-center trial that led to the approval of Kymriah for pediatric ALL. Seattle Children's Group, this is the NCI, this is Fred Hutchinson, and this is Memorial Sloan Kettering, and this is our most recent publication that was just accepted in JCO. So I think the conclusion is that around 50% of people, mostly kids, relapse after having achieved a complete response, an apparent complete response to CAR T cells in ALL. An open question in the field, and something that is close to my heart as a practicing bone marrow transplant physician, is do we still need a bone marrow transplant after achieving a complete response in ALL? And this is controversial. Our experience has been, yes, absolutely, that here are adults with ALL. A lot of these are pediatric. These are adults with ALL. This is not randomized. These are patients who went on to an allogeneic stem cell transplant or not, after initially achieving a complete response to CAR T cells. And so I think the answer is probably yes. Non-Hodgkin lymphoma is a different story, and I would really love to hear if anyone has any ideas about the differences in biology that explain this. But while the complete responses are lower, like I said, about 40% perhaps, the relapse rates are much, much lower than they are in ALL. So if you do achieve a complete response in lymphoma, you are most likely to stay there. And in CLL, as I mentioned, something a little bit close to my heart as well. This is the original, this is an update of the original publication from Penn. This is from Fred Hutch. This is something that we just submitted for review, and this is my paper that is about to be submitted as well. So here what you see is probably a mixture. In fact, what it comes, my sort of sense, although given that this is unpublished as yet, you'll have to take my word for it, that if you do achieve a complete response in CLL, you are very unlikely to relapse. So same as lymphoma, but not the same as ALL. And in myeloma, the problem is, as I sort of mentioned to you, I think is even worse in the sense that the complete response rates, the deep responses are really relatively few, and there's no durability there. So in my mind, I think we still have a lot of work to do on how to advance the field further. There was and still is a lot of enthusiasm, but we need to do better, and we need to think more deeply about what's going on. Solid cancers, this, unfortunately this is the only slide, and you can see how sparse it is. These are many different antigens have been targeted in clinical trials. These are some of the ones that I selected. CEA for colorectal cancer, pretty clear response rates there. HER2, GD2 in neuroblastoma in children, mesothelin and EGFR variant 3, which is, to my mind, the only cancer-specific cell surface antigen that one can therefore target with CAR T cells, and this is in glioblastoma multiforme. So what are the potential mechanisms of failure? I don't sort of mean to put a poll on the field. It's undoubtedly very, very exciting clinical results, but I think in 2020 it's time for us to be very sober and very realistic about what we have achieved and what is yet to be achieved. So this, I took this from a recent review by Crystal Makel, Nature Medicine. And so I think I think we need to think from first principles. What are the potential problems with CAR T cells, or maybe even any other T cell therapy or any other lymphocyte-based therapy? So trafficking, maybe the T cells don't get to where they need to be, for example, in solid tumors. Tumor heterogeneity, I'll show you some data on this in a moment, but since the mechanism of action, CAR T cells are targeted therapy. They're targeted to the surface antigen that they are told to attack. And so if some of your cancer cells don't express the antigen, tumor heterogeneity, as often is the case in solid tumors, then you're not going to kill the tumor. Poor T cell expansion and persistence, I'll come back to this later in this talk, but this has been an observation that has been made recurrently. Different groups, different CAR T cells, different malignancies, that poor T cell expansion and persistence correlates very much with failure to respond. And just as a teaser for the rest of my talk, I'll tell you that we've always thought of it as a primary T cell problem, but it may not be a primary T cell problem. And then impaired T cell function are very much a grab bag of things. What immunosuppressive molecules, cytokines, etc., exist in the tumor or the tumor microenvironment. So I'm going to delve into some details, and some of the references are there as well for you if you're interested. So trafficking, how do we know? What makes us think that trafficking may be an issue? Well, here in each one of these slides, I'll show you clinical data on the left and preclinical data on the right. So responses in hematologic malignancies, where T cells want to go, are generally higher than those in ALL and myeloma, generally higher than those in diffuse large B-cell lymphoma, which often doesn't involve the bone marrow. The few cases of marrow reported, the few cases where solid tumors have been shown to respond to CAR T cells, generally, that's when the solid tumors are metastatic to the bone marrow. And finally, there's a very interesting thread, both clinical and preclinical, thread of data that shows that if you administer CAR T cells locally or loco-regionally rather than intravenously, then you get better responses. This is a New England paper by Christine Brown from the City of Hope, a patient with glioblastoma treated with IL-3 receptor alpha 2 targeted CAR T cells. intracavitary into the tumor itself or intraventricular into the CSF space. And it is not until they started giving cells into the CSF space that they saw resolution of all the patients' sites of disease. And that was persistent for some time, but ultimately we know the patient relapsed. Preclinical data I think is really interesting. Here is an enlargement of that. So this is from Prasad Adusumilli at Memorial Sloan Kettering, again, mouse model, where they administer mesothelin-specific CAR T cells, a 3 times dose, so a low dose, into the pleura of the mice, or a 10-fold higher dose intravenously, or a pleural negative control not targeted to mesothelin. And you can quite clearly see what happens here. But why remains unknown. That has been done also. This is another paper by City of Hope. And here is a paper from our institution from quite a long time ago now that says that if you genetically engineer your T cells to express a chemokine receptor that pushes them to, or at least allows them to follow a chemokine gradient into the relevant area, then that's where they will go and you'll get better responses. So that's been published a number of times. And also more recently in Nature Communications, but by another group showing better responses. So I told you about trafficking. What about tumor heterogeneity? So here's some clinical data and preclinical data. And I think this is really, this is definitely really important. And I remember being in the lab, back when I was running flow myself, and ran flow on the first patient who had a City19 negative relapse, which was shocking to us, but now is very well understood. So in Shannon Ward's paper from the New England Journal of Medicine from 2018, the vast majority of kids treated with CAR T cells directed against City19 and co-stimulated by the co-stimulatory molecule that we favor, which is 4-1BB, as opposed to the City28 co-stimulatory domain that is used by some other groups. The vast majority of the relapses were City19 negative. So one way of thinking of that is the only way for leukemia to escape from this very potent and persistent CAR T cell attack is by losing, downregulating. There are many mechanisms I'll tell you about in a second how they can lose City19. That's also been found by Rebecca Gardner's group from the Seattle Children's Hospital, 7 out of 18 relapses being City19 negative. And that was we never saw that before the age of really potent immunotherapy, and we rarely see that with blinatumomab, which is a bispecific T-cell engaging antibody, although it is seen, but rarely. And this is interestingly, the Memorial Sloan Kettering group, and these are adults mostly, so that's different to these two. Only 4 out of 25 patients who relapsed had City19 negative. And I think the reason is, so adults and children, to be sure, that's a difference, but this is a City28 co-stimulation, and this is 4-1BB co-stimulation, and those aren't esoterica. There are significant differences in the way that the T cells function if you stimulate them with this or that. So the first paper to describe a mechanism for loss of City19 was published by our group. This is from the Children's Hospital of Philadelphia. Andrea Thomas Tinkenenko was the senior author, and it was about alternative splicing. So it turns out that there are probably pre-existing in the patient's cancer, leukemia, various different subclones of ALL, some of which lack, for whatever reason, best known to themselves, lack various domains of City19, and it is not until you exert significant pressure that those come to the fore. I'll enlarge this for you in a second, but suffice it to say that this was built upon by a collaboration between our group as well as this is Elena Orlando from Novartis, who worked with us on a lot of these. And let me show you in larger font what we found here. So this is published in Nature Medicine recently, and here is the City19 molecule. Okay, so it has all these domains and exons, and this is the intracellular domain, these are extracellular domains, and this loop is where the vast majority of CAR19s bind. We know that because most CAR19s are based on an original clone, anti-City19 clone, like an antibody clone, that is called FMC63. Most of them bind here. There is another one that's in use that doesn't bind there. So if I recall correctly, the first patient that we looked at actually is this one, and lacked this domain such that City19 was sort of shed. because it was not anchored to the membrane. And you can see, essentially by sequencing, you can find out what is missing from CD19, and each patient is different. There's no sort of rhyme or reason or pattern as to why this occurs. And it is and leads to basically either truncation or lack of membrane anchoring, or even in some cases simple skipping of the exon that is required to encode that domain. Do the CAR T cells do this actively? I don't think so. We've not studied that directly, but I think it is simply a matter of, and I might be wrong, but it's simply a matter of a variety of different mutants, if you like, pre-existing in the patient and coming to the fore under selective pressure. And so that, I would call this clinical data. There is preclinical data that starts to get at other mechanisms, and I'll show you, as I mentioned in the second half of the talk, additional mechanism. So this is Jacobi from, I think he was at NCI when he published this, myeloid lineage switch. Right, so there are certainly instances, and we know this from clinical practice as well, where ALL sort of relapses as an AML, or vice versa, that's less common. But in this instance, this was really nice mouse work that showed that under the pressure of CAR19, the leukemia in the mouse relapsed not by loss of CD19, but by lineage switching, a sort of program switch to myeloid leukemia that then no longer expressed CD19. And we've seen this in patients as well, particularly in MLL rearranged patients. And this was from my group from a few years ago now, where we simply did something very simple. We just took the blasts, we sorted them into CD19 positive and negative populations, and City19 which is one of my favorite antigens to look at, and did FISH in a patient with a known, we did several patients with known cytogenetic abnormalities, and just very simply found that that the cytogenetic abnormality was present in CD19 negative cells. All right, so let's take a broader view of BALL, and how likely is it that even just at baseline before CAR T-cell therapy, you might have loss of CD19. So this was published from the Children's Hospital of Philadelphia. Vinod Pillai is the senior author here, and they took a whole bunch of BALL cases. On the y-axis is the percentage of CD19 negative, right? So sort of the opposite of what you might imagine, whereas all of these are CD19 positive, but you can start to see when you array them in this fashion that there are certainly many cases that are a little bit CD19 negative, and some cases that are even mostly CD19 negative, which of course we all think of as a canonical B-cell marker. And they did the same for CD22, which of course is also a sort of a B-cell lineage marker. It is of great interest because there are at least two different, well, one approved treatment for CD22 positive disease, which is an antibody-drug conjugate called inotuzumab that works very well. And also we and others have developed anti-CD22 CAR T-cells to deal with the problem of antigen-negative relapse. And you can see that there are patients who have CD19 dim or negative, CD22 dim or negative, and we also increasingly find through clinical experience that if you treat a patient with blinatumomab, they may relapse with a CD19 dim disease that is still CD22 positive, and if you treat them with inotuzumab, they may relapse with a CD22 dim or negative disease that no longer can be treated with CAR T-cells. So a bit of a whack-a-mole sort of thing going on. And so these are all the BALL cases, 628. Here of the CD22 negative cases at baseline, or CD19 dim cases at baseline, so not many, right, as a proportion. But for example, for CD19 dim disease, you're most likely to have hyperdiploid ALL, which is a particular subtype of ALL. And for CD22 disease, you're most likely to have this rearrangement. As a specific example, Philadelphia-like ALL, for example, is very rarely CD19 negative disease, and this is useful because that is otherwise considered a very poor prognosis group, and BCR-ABL, or so-called Philadelphia positive ALL, very rarely such as well. So that's all well and good, and that sort of concludes that part of that sort of second quarter of the overview of me telling you what we and others have observed. Now, what about the poor T-cell expansion and persistence that I alluded to at the beginning? This is when we don't often get results in real time, to be honest, but when we do our pharmacokinetics of CAR T-cells, just like you do therapeutic drug monitoring, if you like, that's how I think of it. If we see a patient who doesn't have a lot of CAR T-cell expansion, and to me that sort of means around 10 or 100,000 copies per microgram of DNA, we start to worry. Typically, of course, that's associated with a very, very simple clinical observation, which is they do or do not have a fever as part of cytokine release syndrome. So here's clinical data, and again, preclinical or correlative data. I think the sort of first observation was from David Porter from our institution, showing that in CLL, patients who have poor expansion and persistence do poorly. This is from the Hutch, showing a similar thing, but also that if you lymphodeplete the patient, if you give chemotherapy, fludarabine, cyclophosphamide, you do enhance the ability of the T cells to expand. And this is data from Novartis via the Children's Hospital, as well as several other pediatric groups, and I'll enlarge that for you in a second, basically showing my point. So, as you can see, the vast majority of children treated with CAR T cells, and this is Kymriah, this is anti-CD19 4-1BB co-stimulated CARs, have a sustained CR or CR with incomplete count recovery. There are a few who have City19 positive or City19 dim relapse. I would say around 5 to 10% of ALLs. And here's the City19 negative relapses that I told you about before. If you compare the pharmacokinetics here and here, they're about the same, except that here you get a truncated follow-up. Often these patients will go into other treatments, but the peak of expansion is about the same. The peak of expansion, and I'll show you more statistics momentarily, is lower in those patients who go on to have City19 positive relapse or who are primarily refractory, for a reason that I'll explain in a moment. And there are some other observations about poor T-cell expansion and persistence. You might ask yourself, well, we're putting in a chimeric protein. We're putting in something into the body that does not exist there. Could it be immunogenic? I have to say that I think the jury is out on that. We haven't found it to be clearly immunogenic, but I know that the Fred Hutchinson group does believe in that very, very well. And here's an example from Cameron Turtle from the Fred Hutch, where they give a first dose and then a second dose of CAR T cells when the first dose has failed. And they find, more often than not, that a second infusion, after initial failure of the first infusion, does not work. And they do have correlative data to show that they have stimulated a cellular immunity to the CAR, which I think is important. Preclinical and correlative data relating to expansion. I'll show you here. So this was a paper from our institution, from Joe Prieta, published in Nature a few years ago. A really, really interesting story. So this was a patient with CLL who was infused with CAR T cells quite a long time ago now, and had nothing happen. And for a long time. And then we gave him a second infusion 70 days later, and you can see a little bit of blips of the CAR T cells. This is quantification of CAR T cells on the y-axis and time over time, and time over the x-axis, and the CAR T cells suddenly explode. And in correlative data that was eventually done, it turned out that all of these cells came from a single cell, because we sequenced the T cell receptor VDJ, and it was a single clone, presumably a single cell. And in fact, we then did lentiviral integration site analysis and found the mechanism for this, which was that the virus had integrated, unbeknownst to us at the time, and certainly not deliberately, integrated inside the TET2 locus, disrupted TET2 function in a patient who knew this, already had a hypomorphic TET2 allele on the other allele, and therefore essentially made a super T cell stem cell memory cell that caused this, and caused the masses of CLL to basically go away and remain away for long term. But this was not a T cell leukemia, because the T cells then went down and have stayed down, and in subsequent analyses of TCR clonality, we found that initial huge expansion, I should say reduction in clonality, now he has polyclonal CAR T cells, although very few. So not a T cell leukemia, and in a sense proof, I think, that a single T cell can eradicate leukemia, albeit in one case. So, and Joe went on to do some other work, published in Nature Medicine in 2018, looking at so if we think that persistence and expansion are important for function and outcome of the patients, what can we do to predict which patients are likely to have that phenotype? And so Joe found in CLL patients that if you have a CD27 positive, CD45RO negative CD8 cells before the T cell production, so it's circulating in your blood, you're a patient who's likely to make good CAR T cells and likely to respond. And that's his data there. And to finish this particular slide, there are many, every week there are additional publications on ways to make CAR T cells better. This is the beauty of synthetic biology. Think up an interesting idea and then test it, and there's a very clear there's a very clear belief that it will be important, but there's a very clear outcome measure. And so I can't even begin to cite all the appropriate studies, but there are many ways to do this. And then finally, impaired T-cell function. So this is nebulous because there's so many different ways of looking at this and so many questions to ask. But some clinical data, and not good quality as yet, but it shows that if you inhibit PD-1, if you block PD-1 after failure of CAR T-cells, you do have some patients that have a good response, a better response. We published a case report on that. I have to say I've done it myself. It doesn't always work. It doesn't usually work even, but it has worked. Meaning if you have CAR T-cells infused into a patient, typically with lymphoma, and it doesn't work, you could try, and of course, well, I'm not supposed to say that since this is CME, but that's definitely something that people have looked at. The City of Hope is looking at just going and selecting cells from the very beginning that are of a more central memory or effector memory phenotype, and infusing those, and they have several trials that are open with that, and some updates on their papers, updates on their trials in the recent ASH meeting. And you could also ask, let's go to a more sort of basic science question: what T-cell characteristics, individual T-cell characteristics even, correlate with response? And so this is a group. I think this is from Boston, basically showing non-responders and responders, and they did a polyfunctional signaling index, essentially analyzing the production of cytokines from single cells in a well using a sort of microfluidic device, and showing an interesting correlation between response and the ability of individual T cells in that patient to make a lot of cytokines. How else can we improve T-cell function? Well, you could maybe think about rational combinations of CAR T-cells with small molecule drugs that we and others have been working on to treat cancer. And so this was published from my group, and then Joe Prieta again at Penn, combining ibrutinib, which is a BTK inhibitor that was developed for the treatment of CLL, with CAR T-cells. Unlike many other anti-cancer drugs, ibrutinib does not inhibit T-cell function, and it may even have some salutary effect. And on the strength of this, we then conducted a clinical trial of combination CAR T-cells and ibrutinib and CLL, which I think worked pretty well. You could also get your CAR T-cells to produce, just to spit out a PD-1 blocking antibody. That's been done. You could knock out PD-1 using CRISPR, and you could inhibit various other you name it, and your favorite immunosuppressive pathway, you could block it in any variety of ways, and that has, and should be tested. So that's the overview section, but a question that occurred to us, and this was probably around, I think about around 4 years ago, when Marco was in my lab. So Marco is now a faculty at Penn. His lab is down the corridor from mine, but at this time he was a postdoc in my lab, and we thought, let's use the new CRISPR technology to do a very, very unbiased CRISPR screen. Let's transduce a B-cell leukemia cell line, NEL6, a sort of standard workhorse cell line, with basically a CRISPR library that encodes. That one was 4 different guide RNAs for every human gene, and then do a stress test and expose those transduced cells to CAR-19 and see what comes up. In theory, if any leukemia survives that stress test, by sequencing that leukemia, we should be able to find out which genes are required or not required by the leukemia to evade CAR T-cell killing. And so the very first experiment isn't even in this. This is a paper that was just accepted for publication, but the first experiment isn't even here because it was so obvious and not that interesting. Because so Marco went back to Italy for Christmas, and I was looking after the cells back then, and had the CAR T cells with the leukemia cells in culture for a month, and they were luciferase positive, and nothing. I couldn't see anything. Eventually, over Christmas, I ran the cells, and there were a few wells with luciferase signal. And then we looked at those particular cells, clones that had grown out, and they all lacked City19. Surprise, surprise, right? And then when we sequenced them, of course, what we found was that they were enriched for guides against City19. So that was the mechanism, right? Not surprising, didn't make it into this paper because it's so not surprising, although it is nice that it does validate what we see in the clinic. So then we did something else, and that's where this story starts. So again, we used a slightly different antiviral CRISPR library. It's called Brunello. I think there are 6 different guides to every gene. We used the same cell line, um-lmp. We transduced, we puremized and selected, we exposed to CART19, but this time, partly, I have to say, partly inspired by what people, sort of in the intermediate period between our first results and this, what people were doing with drugs or actually even immunotherapy in the checkpoint field. We did a short-term assay only to look for what sort of a short-term exposure to CAR T cells. Does it tell us anything interesting? So what we found is this. We did this very short-term exposure, took out the leukemia cells and looked to see which ones had enriched or depleted guides or genes. And this is what we found. Upon exposure to, so here's the y-axis CAR T cells, control cells. This tells you that they're equivalent. Above the diagonal tells you that these genes are enriched in leukemia exposed to CAR T cells, and below are depleted upon exposure to CAR T cells. So what we found was that these generally belong, at least the most outstanding ones, belong to a single pathway, which was the extrinsic apoptosis pathway. Caspase-8, FAD, BID, BRCA2, TRAF2, CFLAR, all of these belong to the same pathway, and you can see it here. This pathway analysis shows that the vast majority, by a very, very high margin, of genes that we came up with were belong to the apoptosis pathway, specifically extrinsic. And this was surprising because we think that CAR T cells or T cells work generally by different granzyme and interferon and all that sort of thing. Nonetheless, we pursued it. Oh, and I should say, when Marco became independent, then Nathan Singh, who was a postdoc, I beg your pardon, a hem-onc fellow, then worked on this, completed it, and recently moved to WashU to start his own lab there as well. So, and so this is what we found when we wanted to validate these results. We selected BID and FAD. We didn't validate all of those genes, but we validated some of them. But here, what I'm going to show you here is FAD in blue, BID in red, and wild type in black, referring to um-lmp cells, so ALL cells, that we then CRISPRed specifically to lack BID or FAD or DID CRISPR. And we exposed them to CAR T cells, CART19, for several hours, a long-term assay. And you can see that there is, this is after 24 hours of exposure, etc., and this is percent-specific killing. And here you can see that killing of leukemia increases over time in standard wild-type leukemia. But the lack of ability to respond to extrinsic apoptosis-inducing signals that, via FAD or BID diminished the ability of that leukemia to be killed by CAR T cells, and that maybe even accelerated over time, which is something I'll get back to in 1 second. We confirmed it in using CAR22. We confirmed it in lymphoma as well. And so that observation sort of, really at the time we were thinking, what could this possibly mean? Because we really only exposed the leukemia to the T cells for such a short time, and we know that in patients that sort of, if you like, the area under the curve leads us out to at least 2 weeks, if not longer. So what could this possibly mean? We then did the in vivo experiment in mice, and here, let me take you through it. This is the FAD um-lmp, this is the BID um-lmp, same controls. We engraft immunodeficient mice with wild-type um-lmp in gray that are treated with control T cells, and they die because there's no anti-leukemic effect. We engraft them with wild-type um-lmp and treat them with CAR19, and they survive longer. This is an aggressive leukemia, and we deliberately underpowered the system. We certainly can cure mice using CAR19, but we sought to underpower this in order to bring out potential differences. And here we had the same leukemia where we had previously CRISPRed out FAD or BID on the right here, FAD here, BID here. And we treated them with control cells or CAR19, and there was no difference. So it is striking, I think, that in vitro, these are maybe, they're significant, statistically significant, but kind of marginal biologically. But the effects in vivo are much more impressive than they are in vitro. Perhaps because the in vivo experiment allows you to explore kinetics and additional downstream adaptations, which is what I'll talk to you about. So in order to model that in vitro, we did this, bear with me, this is rather complicated, but I'll take you through it step by step. So we did an initial exposure of CART19, these blue circles here, CART19 here, CART19 here, same CART19, but we exposed them either to wild-type NALM6 or to NALM6 lacking BID. And after a short or long-term exposure, short is 5 days co-culture, long exposure is 15 days co-culture. After that, we then sorted the CAR T cells and re-exposed them, but this time just to wild-type NALM6 that we know they can kill very well. And then we tested the effective function. So here's a short-term incubation. So CAR T cells exposed to wild-type or to knockout NALM6 for a short time, and then we look at the ability of the CAR T cells to proliferate upon exposure to the same antigen, nothing wrong with City19, upon re-exposure to the NALM6, and there's no problem with either of these, CAR19 exposed to wild-type NALM6 or CAR19 exposed to BID knockout, apoptosis incompetent, if you like, NALM6, they're still able to proliferate. But when we do that after a 15-day culture, so they're together for 15 days, if you then sort out the CAR19 that had been exposed to wild-type NALM6, they're still able to proliferate in response to wild-type NALM6. But if you sort out the CAR T cells that had been exposed for a long time to this death-incompetent leukemia, they don't proliferate. Same is here, we looked at cytokines, so short incubation and then re-exposure to a wild-type NALM6, no difference in the ability of cytokines to be produced, interferon gamma, TNF, MIP1 alpha, but after a long exposure, followed by sorting and re-exposure now to a wild-type NALM6, you see that the cells that had been exposed for a long time to a death-incompetent leukemia don't have the ability to, this is a different scale too, don't have the ability to produce these cytokines. And of course, we need to test their ability to kill that second exposure to leukemia. And this is, again, after the short incubation, you can see that this is NALM6 quantification. So there's, here we have the killing of NALM6 at 48 hours. So slight reduction in the ability. So if you have your cells together, the CAR T cells with the wild-type NALM6 or the killing incompetent NALM6, sort them, re-expose them for a short time to NALM6. And you can see that initially, after a 48-hour retest of their killing ability, you know, they can proliferate, they can produce cytokines. They're not as good at killing initially, but by the time you let them go for 120 hours, they're killing leukemia. But if you go to those, if you take those cells, and I'm belaboring this because it's rather complicated, if you take the cells after prolonged exposure to the death-incompetent NALM6, they produce less cytokines, they don't proliferate, they don't kill, and they still don't kill after 120 hours. So they're pretty exhausted. So we did RNA-seq and PCA, and you can see, not surprisingly, that here are your resting cells, T cells. Here are your T cells exposed to wild-type NALM6. Here are your T cells exposed to a BID knockout NALM6, and they're different. How are they different? Well, they show differences in various dysfunction-associated genes, upregulated. So on the right, it's upregulated in our CAR T cells that are exposed to death-defying, if you like, leukemia, CTLA-4, CD200, BTLA, TOX2, which was published in 2 or 3 back-to-back Nature papers recently as an important transcription factor of exhaustion, TIGIT, TIM3, etc., etc. And conversely, lower expression in these exhausted T cells of several other important markers, Alzheimer's receptor, KLF2, etc., etc. So, and then we did ATAC-seq, and the reason we did so is to at least try and get at the mechanism, and of course the fact that we did not, in the previous slide I showed you that we couldn't really reverse this exhaustion after letting the T cells be away from the death-defying NALM6, and so we wanted to find out why. And so this is ATAC-seq of resting T cells, CAR T cells exposed to the wild-type NALM6, CAR T cells exposed to the BID knockout, death-defying NALM6, if you like, and this is where the major difference occurs. And then if you look at global promoter access, here are some, let me take you through it. This is going a little bit low, but let me take you through it. So the top is the KLF2 gene. then we have PRDM1, and then we have TOX2. So the first one is a more T-cell, pro-T-cell function, if you like. The bottom two are T-cell exhaustion. And in each panel, the first track, the top track, is resting CAR T-cells. The second or middle track is T-cells exposed to wild-type NALM6, and the bottom track is CAR T-cells that are exposed to a BID knockout, death-defying NALM6. And you can see clear differences, I think, so the gray shading indicates for you, for example, that the KLF2 promoter site is open in resting or wild-type exposed CAR T-cells, but closed, more closed at least, in BID knockout. Conversely, CAR T-cells that have been exposed to BID knockout NALM6, the loci of promoters of genes that are involved in exhaustion are more open than in the controls. And so there is an epigenetic phenomenon there as well that may explain why it is progressive exhaustion and potentially irreversible exhaustion that occurs when CAR T cells encounter leukemia that they can't immediately kill. That's all well and good. That's all mouse stuff and it's in vitro stuff. So what about patients? So these are two patients, two pediatric patients treated with CAR T cells, CAR-19, ALL, one of whom responded, as 90% of them respond, and one of whom did not respond. They both had City19 expression. And in this, so this is the 10x genomics platform. Here we put the two products together. These are the CAR T cells. So we took the CAR T cells, sorted them from the infusion product. We had leftover cells, sorted them from the infusion product, put them back to, sequenced them, single cell sequencing, and put them back together. And we colored them here for your delectation as the responder, the non-responder in the CAR T cells at infusion, and the CAR T cells at expansion. So you can see there's numerically not really much difference. If you just compare, we put the same number of cells. There's not much difference in CAR T cell quantity in the responder and the non-responder. And this is at peak expansion, which typically happens around day 10 after infusion. And then we looked for the expression of exhaustion-related genes. We made a list of exhaustion-related genes, and the first question was, is there a difference in exhaustion-related genes in the infusion product? Can you predict that the T cells that you've made are exhausted already, and maybe there's no point in fusing them? And the answer is no. I'll show you in a second another way of looking at this data. Doesn't seem to be much different. I should tell you that each dot here is a single cell, and as the color deepens, that means more and more and more exhaustion-related genes, right? So we sort of did this. If the lightest color is no exhaustion-related genes, 1, 2, 3, or 4 or more exhaustion-related genes. So the more deeply colored cells are perhaps more exhausted. So at baseline, no difference. But at the peak of expansion, where numerically there's not been a change in the number, there's no difference. At that time point, at least, there's not a great difference in the CAR T cell quantification between these patients. You can see, I think, that these cells that are from the non-responder are more deeply colored and have generally more exhausted cells. Same as what we found in vitro. And this is another way of looking at the same data. Again, same scale, so no markers, 1, 2, 3, 4 or more in the infusion product. Not a great difference there, but I think you can see that at the time of peak expansion, there's certainly more of these exhaustion-related gene expression in the patient that was a non-responder. And so then we went to we looked at two clinical cohorts. I'm showing you one of them, but we validated it in a second one as well. This is again from pediatric patients on a multi-center study treated with the same CAR-19 drug, and we looked for. We made a list of extrinsic apoptosis genes, and we made a list of intrinsic apoptosis genes as a control. And I'm not showing the control; it's in the paper, but I'm showing you the extrinsic apoptosis. And there was a difference. The reason this is not like what you're used to seeing from a heat map is because it's not one particular gene. It's not just FAD or just BID. It's any one of these, or several of them, in an individual patient who's a non-responder, who has lower expression of these genes that are required for killing. Conceivably, you could also have higher expression of genes that resist killing, which is what we found in our screen, but not necessarily in our patients. For example, this particular individual, just to take you through it, this is one individual who has much lower expression than most other ones of RIPK1, of TNFRS of 10A, TRAD, all of these guys, caspase-8, caspase-10, RIPK3, all of these that are genes that are required for response to killing. And his or her neighbor looks pretty similar, though there are other patients who don't look like that. But generally speaking, across the board, that's what we found. And then we made a score, so patients with a high score versus patients with a low score. And you can see that if you, and this is the death receptor gene signature score. So this is to sort of validate our data. It is sort of definitely statistical mishmashing. But nonetheless, in the non-responder patients, the vast majority of those had very low death response signature, whereas the vast majority of complete responders did not have that problem. And this was, of course, statistically significant. I should tell you, this came from marrows at baseline before treatment, blood. We selected patients whose marrow RNA-seq we could get, and we also knew their blast percentage, and so we corrected this for blast percentage, since some patients have 90% blast, some patients have 90% blast, and when you just do, this was bulk RNA sequencing, there's a possibility of washing that out, so this was corrected for blast percentage. And this is the same data set. Here is the expansion, right? So this is pharmacokinetics. This is CAR T-cell quantification on the y-axis and time after infusion on the x-axis. And each individual line is an individual patient, and in black are the in black are the high score, which is generally the complete responders, and in red are the low score. So patients whose leukemia appears less competent to be killed by the extrinsic apoptosis pathway, but not by the intrinsic apoptosis pathway or any other pathway. So, and you can see, and here I've just enlarged it. This is an inset from early time points. You can see that this goes out to day 1000. This only goes down to day 80. So this is an enlargement of this inset for the sake of clarity. And what you can see here, roughly, I think, is that here is the peak expansion of our responders. As I showed you before, this is reproducible time and time again. Responders have a very good expansion. Non-responders do not. But here, we didn't divide it by responders and non-responders. We divided it by those with a high or low extrinsic apoptosis score. And critically, here is the survival. So overall survival is shown in panel E, where we have all subjects in gray, and then when you dig deeper, you can see the subjects who had a high score did very well, patients who had a low score did very poorly. And in experiments that I didn't show you but are in the paper, we of course confirmed that this had nothing to do with chemotherapy responsiveness, because this pathway is not required for chemotherapy responsiveness. So to summarize that, I'm pretty much done. To summarize that, this is the model that we propose, that if you have a death receptor impaired ALL, maybe also lymphoma, we didn't do that much with lymphoma, but let's stick to ALL for now. If you have a death receptor impaired ALL, it doesn't have to be bit or fat, it could be a variety of other different things, but we mostly studied bit and fat, that's in red, compared to a death receptor intact ALL. And you expose both to CAR T cells. The death receptor intact ALL is killed, and that allows the CAR T cells to survive and do what they like doing. But the death receptor incompetent ALL persists, at least a little bit, probably long enough to induce, through a dominant negative effect, induce exhaustion in the CAR T cells, and thereby mediate a much more dramatic T cell dysfunction than you would have initially thought would occur from the short-term in vitro killing results that I showed you. And so essentially you sort of get a dominant negative effect that keeps pushing the CAR T cells further and further and further into exhaustion until a point where maybe they're even unrescuable. And that's schematized there for you here, but you just saw the data. Clinical outcomes, CAR T cell expansion, high death receptor score, low death receptor score, and then survival. And I think that this might explain these results. I certainly, and I think most of us always think it is a primary T cell problem, but it may be a primary leukemia problem. And those of course may interact, because the T cells that you make come from a patient whose leukemia is deficient in killing. But having said that, we did compare the T cells at infusion, so the manufacturing product, and found that there were no differences in the product. So when we take T cells out with apheresis from the patients and we manufacture them, they spend a long time being stimulated and away from leukemia, and they sort of reach probably, and possibly based on our data, though I think it needs to be deepened further, the T cells are somewhat, they recover. The CAR T cells recover. But then you put them back into a patient whose leukemia is death receptor defying, and then they get exhausted. And so at least, maybe both mechanisms are true. I'm sure both mechanisms are true, but at least I think the novelty of this is that it shows a dominant negative effect of the ALL on the T cell. And if you like to think a little bit further, before the days of living drugs, what situation can you think of where a patient's disease characteristics impact the pharmacokinetics of the drug you give them? Not many. So to me, that was interesting. It was one of the most interesting things about this. And so to summarize very, very briefly, I told you about problems and potential ways of improving CAR T cell therapy in 2020 that may deal with tumor heterogeneity, with trafficking, that may deal with T cell expansion and persistence, and that may potentially deal with specific T cell suppression. And so finally, acknowledgements, of course. So Marco now has his own lab where half of these people work, half the other people work in my lab, and Carl June and Noel Fry, Shelley Berger are senior scientists or clinicians at Penn, collaborators at the Children's Hospital of Philadelphia, Novartis, and then funding sources. And thank you to you for your attention.
We have time for a few questions. Chris?
If your model of T cell dysfunction is accurate, then you predict that the endogenous B cells would initially tank but then recover as T cell dysfunction gets established. Is that what happens in those patients?
Yes, generally speaking. Yes. So actually we were talking about this last night. So we can quantify CAR T cells through a variety of flow or qPCR, but to me what matters is what are the endogenous B cells telling you about the function of the T cells. And yes, generally it's binary. So CAR T cells yes, B cells no, CAR T cells no, B cells yes. But what makes it of course complicated is in situations, practically in situations where you have relapse disease, there's not a lot of time to observe those things occur, and the patients are often going on to immediately next line therapy. Bruce?
That was a really nice talk. Can you tell us if the extrinsic apoptosis pathway is critical? In vitro, if you continue to put in new antigen, does that simulate it?
Yes, it does. Exactly. So you don't necessarily need. To me, it's all about antigen persistence, right? Not surprising. That's how exhaustion. We know that's how T cell exhaustion occurs. Yes, if we did that, and it's in the paper. If you just forget all about the knockout and the extrinsic apoptosis, you just keep feeding the T cells, the CAR T cells with wild type leukemia. Exactly the same thing happens.
And then when the normal B cells come back, why doesn't that happen?
Why doesn't normal B cell recovery? So normal B cell recovery occurs once CAR T cells have gone. Anyway, and I imagine there might be additional mechanisms that we haven't studied, but that absolutely bear studying. What else is on the leukemia surface that mediates this? Yes.
Can this exhaustion be overcome by repeated CAR T doses?
Theoretically, yes. We didn't study that. Theoretically, if that is the sole mechanism, then just giving fresh CAR T cells should do the trick. We haven't done it preclinically, and we haven't really systematically done it clinically either. What I fear, I have to tell you, is that in some situations that the patient may be telling you that so you've kind of selected leukemia. Maybe what I showed you with the bulk RNA-seq, of course, is bulk, right? So there are some clones that are resistant, and some that are not resistant to killing via this pathway, and I sort of fear that maybe when we do that, we'll find that we don't succeed, not because the CAR T cells aren't fresh, but because most of the leukemia is now resistant to.
Is there any association between patients' age, the duration of their prior therapy, or their type of prior therapy, and the likelihood of their T-cell product becoming exhausted?
We haven't done that work yet. I think you need a lot of patients, we haven't done it. I'm sorry, you had a question?
Right. Yeah, so I was wondering, how common are these mutations you find, not necessarily in TET2, but is this sort of a more common feature of CAR T cells? Do you see clonality frequently? In other words, is it more than just a CAR and sort of putting these in with lentiviruses, we're targeting unknowns to us?
Yeah.
Other pathways
Mhm.
that cooperate a
That's
lot?
a
Or
very
rare?
good question. We just published in JCI, maybe in the last month or so, a bigger, at 30 CLL patients, where really it was just a survey of lentiviral integration sites. And no, with the exception of sort of the exceptional case reports where something notable happened, we don't see clonality. We see, depending on the number of T cells that are there, of course. And so there doesn't seem to be a selection. At the peak of expansion, there are usually many different clones based on LVI, lentiviral integration site. And later on, it does appear more like a clonal, but that's probably only because you have very, very few T cells.
Early in your talk, when you were talking about the CD19 negative relapse in ALL,
Yes.
I noticed when you looked at the CD19 molecule, all of them had persistence of the number 2 epitope. Is that because the CD19 doesn't react with the number 2 epitope?
I suspect so, yeah.
Oh, okay.
I think it's because, so anything upstream in this type of protein of the site that is required for binding by the CAR gives the leukemia no proliferative growth advantage. Escape advantage, I should say.
Yeah.
Okay, let's thank Saar again.